Today, we're cracking the code on a really common type of cancer. We're going to explore how a diagnosis that sounds safe can sometimes hide a dangerous secret, and how digging into its genetic blueprint could completely change the game for millions of people. For years, doctors have looked at one specific type of breast cancer as, the safest one, the one with the best odds. But what if that's a huge oversimplification? What if, hiding inside that safe diagnosis, there's actually a much more aggressive disease lurking for some people? Well, that is the puzzle we are diving into. So the cancer right at the heart of this mystery is called Luminal A. On paper, it looks like a good cancer. But as you're about to see, when you look at its genetics, things get a whole lot more complicated. Look, Luminal A totally earned its safe reputation for some pretty solid reasons. As you can see, it's the most common kind, making up about half of all cases. And because it's fueled by hormones, it usually responds really well to therapy. Plus, it's typically slow-growing. All these things add up to a very good prognosis for most patients. But here's the catch. Not everyone with this diagnosis gets that same good outcome. And the reason is this tricky thing called molecular heterogeneity. The best way to think about it is like this. You could have a dozen cars that are all the same make and model, right? But under the hood, one might have a standard engine while another has a souped-up racing engine. Genetically, these tumors can be just as different, which makes it almost impossible to predict which ones are going to suddenly hit the gas and turn aggressive. So how do you even begin to solve a puzzle like this? Well, researchers basically had to become genetic detectives, using a really powerful set of tools to crack the code hidden deep inside these cancer cells. To get it done, they used what's called a systems biology approach, which is what you're seeing mapped out here. I know, it looks super complicated, but just think of it as a forensic investigation. First, you gather the evidence, the raw genetic data. Then, you analyze it to find out what's different or unusual. And finally, you piece it all together to understand who's in charge, to figure out the tumor's command structure.
Okay, so here's the breakdown of how that worked. Step 1, they collected genetic data from 43 tumors, but they also took healthy tissue from the same patient to have a perfect comparison. Then they sifted through all that data to find the genes that were, acting weird in the cancer cells. After that, they mapped out how these rogue genes were all interacting with each other. And finally, they zoomed way in to find the master regulators, the real ringleaders driving the whole operation.
So how many genes did they find that were behaving abnormally? Get this. An incredible 1,999. That's almost 2,000 genes that were either in overdrive or totally shut down in the tumor cells compared to the normal ones. This isn't just a small tweak, it's a massive, system-wide hijacking of the cell's genetic programming.
Finding nearly 2,000 altered genes is one thing, but the really crucial next step is to figure out, what are they all doing? What kind of chaos are they actually causing inside that cell?
And this is what all that genetic chaos looks like. It's called a volcano plot, and every single dot you see is a gene. Just look at that eruption. The orange dots flying up to the right, those are genes the cancer has kicked into hyperdrive. And the green dots over on the left, those are the genes the cancer has slammed the brakes on. This is a complete and total genetic rewiring.
So, and this is the most important part, these thousands of genetic changes are not random. They are systematically hijacking the cell's core operating instructions, what scientists call pathways. These pathways control literally everything, from whether a cell grows to whether it knows when to self-destruct.
Here's a perfect example. What you're looking at is a map of the cell's division process. It's called the cell cycle pathway. See all those parts highlighted in red? Those are genes that the researchers found were stuck in the on position. It's like the accelerator pedal for cell division is completely stuck to the floor. The cancer has just floored it.
And it wasn't just cell division. The investigation showed that this disruption was happening everywhere. The cancer cells were basically learning to ignore the natural signals that tell a damaged cell to self-destruct. They were rewiring their own metabolism to get more fuel. And they were pumping out these damaging molecules, creating a really toxic, unstable environment for themselves to thrive in.
So with all this chaos going on, you have to ask the question, who's in charge? Who were the masterminds pulling the strings and orchestrating this whole cellular takeover? This is where the researchers finally identified the key ringleaders. To find them, they were looking for something called transcription factors. Now, the absolute best way to think about these is as master switches. A single one of these transcription factors can flip a switch and turn hundreds, even thousands, of other genes on or off all at once. They're the ultimate command and control molecules inside a cell.
And here they are, the culprits. The analysis zeroed in on several of these key master switches. A couple were messing with metabolism to feed the tumor, but I want you to focus on these last three: SOX2, TLX1, and PTTG1. This is where the researchers found the smoking gun. High activity in these specific master switches is a major red flag. It's directly linked to poorer patient survival. This is the key. This is how you can finally spot the high-risk patients who've been hiding in plain sight within that safe group.
So what does all of this mean for actual patients? Well, identifying these ringleaders and their weak spots isn't just a cool science experiment. It gives us a brand new blueprint for how to treat this disease.
This research really points toward a huge shift in strategy. The old way was to treat pretty much all luminal A cancers the same way, kind of a one-size-fits-all approach. But the new approach this work makes possible? That's true precision therapy. You analyze a person's individual tumor, see which specific master switches are flipped on, and then you pick a treatment that is designed to shut that exact switch off.
And here's the part that is just so incredibly exciting. The solution might already be sitting in our medicine cabinets. The analysis actually identified existing, FDA-approved drugs that could be repurposed to fight this cancer. For example, a drug for autoimmune diseases called etanercept, well, it turns out it targets PTTG1, one of those key red flag regulators. This is a huge deal because it could fast-track new treatments using drugs we already know are safe for people to use.
You know, the study's authors really put it perfectly themselves. They said their work provides a foundation, a roadmap for figuring out who is truly at high risk, and for optimizing their treatment with incredible precision. It's all about moving past that one-size-fits-all label and treating the cancer that's unique to the individual.
Which leaves us with a final, really powerful question. This study was focused on just one subtype of breast cancer, but the method, the approach they used is universal. So could this kind of deep dive into the unique genetic code of every single tumor be the ultimate key to unlocking truly personalized and way more effective medicine for all cancers? Based on what we've seen here, the answer looks like a resounding yes.